So a disaccharide is just two monosaccharides that are connected by something called a glycosidic bond. So for example, this is a disaccharide of glucose and another glucose, and the glycosidic bond in this case is formed between the number 1 carbon of this glucose and the number 4 carbon of this glucose. And the bond forms when basically there's a condensation reaction between the OH group of this glucose in this case, and then the alcohol group of this glucose, and you end up with this kind of bond here, where the number 1 and number 4 carbons are linked by an oxygen atom. And in this case now the hemiacetyl group that was the number 1 carbon is now a full acetyl group, and in this case the anomeric carbon of the other glucose doesn't participate, so it's still the same configuration, but it can be in either alpha or beta afterwards. So sometimes it's drawn like this, where it's not necessarily clear or not necessarily important whether it's in alpha or beta. And then the full name for this whole complex is alpha D glucopyranosyl 1,4-D glucopyranose. So again, the carbon or the monosaccharide that's linked to its anomeric carbon is the one listed first, and it's always one of the anomeric carbons that forms at least one side of the disaccharide bond. Sometimes you can have a connection between two anomeric carbons, but in this case it's not. So the one that has the anomeric carbon connected is the one that's named first, so it's alpha D glucopyranosyl, so it's still in its alpha configuration because the oxygen is still below the plane of the ring, and the number 6 carbon is above the plane of the ring, and it's still in its D arrangement, and so it's in a ring, so we call it pyranosyl. And then the number 1 carbon is linked to the number 4 carbon of the second monosaccharide, so it's 1 arrow 4, and then the second glucose is also D, but again it can be alpha or beta, so we leave that out, and so it's D glucopyranose. So that's the whole long name of this disaccharide, alpha D glucopyranosyl 1,4-D glucopyranose. But of course that's a lot to say, so a lot of disaccharides just have common names like maltose. So this is a molecule of maltose. And these are just other examples. This is lactose. So lactose is a disaccharide. It's a sugar that's common in milk. That's where the name comes from. And it is a disaccharide of galactose and glucose. So it's beta D galactopyranosyl 1,4 beta D glucopyranose. So in this case, the galactose is in its beta configuration, and the glucose is in its beta configuration, so they both are beta. And then again, it's in this case it happens to also be connected from the 1 carbon to the 4 carbon, so we say it's 1,4 beta D. And then this can be abbreviated, so in this case gal is short for galactose and GLC is short for glucose. And then again the beta 1,4 refers to the fact that they're both in the beta configuration and they're connected from the 1 to the 4 carbon. Here's another one. This is sucrose. Sucrose is probably what you have in your kitchen and buy at the grocery store in big 5-pound bags, and it is a disaccharide of fructose and glucose. So and they're both in this case connected at their anomeric carbons, so it can be written either way. You can write it as beta D fructofuranosyl. So fructose is a ketose, so it's in its furanose form. And then in this case alpha D glucopyranoside, and then its abbreviation can either be this way. So fructose 2 beta. So you're talking about from the number 2 carbon of fructose, which is in its beta configuration, to the number 1 carbon of glucose, which is in its alpha configuration. So you can write it as fructose 2 beta alpha 1 glucose, but and this little double-headed arrow means that they're both connected at their anomeric carbon, and you can write it the other way and it means the same thing. Again, though, this sucrose is just a much easier name to say, so most of these disaccharides have common names. And then here's another one where now it's again a disaccharide of 2 glucose residues, and they're both in the alpha configuration. Its common name is trehalose, but they're connected at the number 1 carbon, the anomeric carbon. They're both in the alpha configuration, and so the other one is just sort of drawn upside down to indicate that again, you still have the oxygen sort of underneath. But in reality, of course, this is not what the bonds actually look like. They aren't curved like this, but just to make them easier to see, they're drawn this way. Also, by the way, these sugars, and actually this was true with some of these monosaccharides as well, are being drawn when they're in their ring structure like this, in what's called a Hawthorne projection, which just means that the ring is depicted in sort of 3D. So you have the bonds that are sort of closer to you drawn a little bit thicker than the bonds that are further away. And then it also reflects the fact that if a hydrogen or an OH is drawn this way, it means it's below the plane of the ring, and OH up here means it's above the plane of the ring. So that's where that comes from. Now, again, we said that sugars can be reducing or non-reducing, or all monosaccharides are reducing sugars. And then if you have a disaccharide in which one of the monosaccharides, or one of the residues, has a free anomeric carbon, then it would be a reducing sugar. So lactose, for example, is a reducing sugar because it has this carbon over here, which is free. In other words, it's not bound to anything, and so it's free to participate or as a reducing agent. Whereas sucrose is a non-reducing sugar because both of its anomeric carbons, the number 1 carbon of glucose, number 2 carbon of fructose, are connected to each other, and so can't participate in a reduction. So sucrose and trehalose also would be non-reducing sugars. That's all that means. And so that affects kind of how they can be detected, but it's also just another way of classifying disaccharides. So again, disaccharide is just 2 monosaccharides chained together. You can also, of course, have trisaccharides and tetrasaccharides and so on. But usually after 2, we start just calling them polysaccharides. So polysaccharide is just something, just a carbohydrate with more than 2 monosaccharides. And there can be basically 2 kinds of monosaccharide, or 4 kinds, broadly speaking. So they can either be all made of the same monosaccharide, in which case they would be Homo Polysaccharides. So these are Homo Polysaccharides here. Every one of these little residues is exactly the same monosaccharide. Or they can be hetero polysaccharides, in which case the monosaccharides that make up the polysaccharide can be different. And sometimes there can be a repeating pattern where you can see here we've got this one has the same 2 monomers, the same disaccharides essentially linked together one after the other, or not. They can be multiple, 3 or more different kinds of monosaccharides that are linked together in a non-repeating pattern. And then polysaccharides can be either branched or unbranched. So the simpler ones are the unbranched state, where you have just a linear chain of monosaccharides that are linked end to end. The linkages don't always have to be the same, especially if it's a heteropolysaccharide. So you can have different types of linkages, but each monosaccharide is only linked to 2 others. And then a branched polysaccharide is one in which one or more of the monosaccharides is linked to more than one or more than two monosaccharide chains. So here, this is a branched Homo Polysaccharide because all the monosaccharides are the same, but you have one branch point here. So this monosaccharide has three chains attached to it. Over here, this is a branched heteropolysaccharide because again, you have at least three different kinds of monosaccharides, and this one is connected to three other branches. So again, that's just a way of differentiating different types. Now, this is one of another important difference between one thing that makes carbohydrates different from the other kinds of polymers we talked about. So proteins, for example, which are chains of amino acids, are always unbranched. There's no such thing as a branched protein unless you count like disulfide bridges. And then same goes for nucleic acids. A chain of nucleotides is always a linear chain. There's no such thing as branched nucleic acids. But polysaccharides can form branched chains. And then another important thing that makes polysaccharides different is that they don't have a defined length. That's not true, again, for protein. So every protein, by definition, has a specified number of amino acids that it's made of. Same is generally true of nucleic acids. So if you're talking about an mRNA or a gene, it's defined by a certain number of nucleotides that is a certain number, a certain length. Whereas polysaccharides can be a variable length. So they can be added to or taken away from, and it's still essentially the same polysaccharide. So these are just some examples of different types of polysaccharides. They generally serve two functions. They are either used as storage or they are used as structural elements. So these are examples of storage polysaccharides. In plants use a lot of carbohydrates because, of course, remember, they're the ones that actually make carbohydrates. So polysaccharides in plants, many of them are in the form of what we call starch. So most of the energy storage in plants is starch. And starch is made up of two different types of polysaccharides. This one up here is called amylose. It's basically just an unbranched chain of glucose that are connected in a 1-to-4 glycoside linkage. Or another common type of starch is amylopectin. So amylopectin is a branched chain of glucose residues that are linked either 1-to-4, and then the branches are linked 1-to-6, and then the branches show up about every 24 to 30 residues. But again, it's not defined. And then in animals, so the storage form of glucose in your body is mostly in the form of glycogen, and glycogen actually looks a lot like amylopectin. So again, it's a branched polymer of glucose, which are 1,4 linked with 1,6 branches, alpha 1,6 branches, but they're just closer together. So the branches are about 8 to 12 residues in glycogen, and they're about 24 to 30 residues apart in amylopectin. And then another example, Dextran is a common storage polysaccharide for a lot of single-cell organisms like yeast and bacteria. Again, it's another branched polymer, glucose. So again, if you're noticing a theme, glucose is is used by a lot of organisms for energy. In fact, we'll talk about how glucose is metabolized later on. And then this is just what a typical starch molecule might look like. So it's a complex of amylopectin sugars with amylose kind of intertwined with it. So you can have complexes of multiple polysaccharides grouped together. One thing, again, we still talk about reducing and non-reducing ends. So every polysaccharide is a chain, and so in this case, amylose and amylopectin both have one end that's reducing and one end that's non-reducing. But with amylopectin, there's only one point where it's reducing, whereas all of the other ends of amylopectin are non-reducing because they're not at the anomeric carbon. But that's not true of all polysaccharides, so it just depends on how they are connected. And then a lot of organisms use polysaccharides as structural components of their cells or their tissues. So plants, for example, have this stuff called cellulose. And cellulose is similar to amylose in that it is an unbranched polymer of glucose, but it's connected in a beta-1,4 glycoside bond. So remember, with amylose, it's connected in an alpha-1,4. That may not seem like a big difference, but the beta-1,4, meaning that the first carbon in each dimer here, or the first sugar in each dimer, is in the beta configuration, simply means that the two of them have a slightly different structure, and this allows these hydrogen bonds to form between adjacent units, which is really important because it stabilizes the structure and makes it very sturdy. And so cellulose, in fact, is the main structural component of most plants. It makes up what's called the cell wall, which is the structure that surrounds the cell and gives it physical shape. And a lot of extracellular tissue in plants is made of cellulose. In fact, most wood, most of the rigid parts of wood is made of cellulose. If you have a piece of paper in front of you, that piece of paper is mostly made of cellulose. Now, you might think, well, if cellulose is basically just the same structure as amylose, why is it that we can eat the starchy parts of plants? So, things like potatoes and for example, is made mostly of starch. But we can't eat cellulose, and it's simply because we just don't have enzymes that can break down cellulose into their components. We do have enzymes that can break down amylose and amylopectin, probably because it's very similar to glycogen, but we don't have enzymes that can break down cellulose. In fact, no animals have the ability to break down cellulose, so that's why you can eat, again, potatoes, but you can't eat trees. But there are some organisms that do have enzymes that can break down cellulose. So, for example, this little eukaryote, it's a little single-celled eukaryote, has an enzyme called endocellulase that does take cellulose and break it down into glucose, and then can actually use that for energy. So these guys can eat things like wood and paper and on their own. And then there are some animals, like these termites, that themselves don't have the enzyme to break down cellulose. Again, no animals have it, but they have these little guys living in their digestive system. So the reason the termites are able to eat wood is because they have these little endosymbiotic organisms that break down cellulose for them. Animals do have, though, some structural polysaccharides. Chitin is a good example. So all arthropods, so that means that includes insects, it also includes things like crustaceans, have basically an outer shell or an exoskeleton, and that exoskeleton, the structural part anyway, is supported by this stuff called chitin. And chitin is basically an unbranched polymer of polyacetylglucosamine. So this stuff is embedded in a matrix that includes proteins and lipids that kind of support the outer structure of the whole animal. Now it's secreted from the skin, so as the animal grows, it has to be shed periodically. So that's why, and then regrown. That's why insects and crustaceans go through molting phases where they get rid of that exoskeleton and grow a new one. And then in animals and in some other multicelled organisms, these structures called Glycosaminoglycans are important parts of extracellular matrix. These are just some examples. So hyaluronate is a really big polysaccharide consisting of these glucose, glucosamine, and N-acetylglucosamine chains. So this makes these big kind of slimy substances. So anywhere you need a sort of thick fluid in the body, you'll find this stuff. So the vitreous humor, for example, is the fluid inside your eyeball. It's what gives your eyeball its shape and structure. And then synovial fluid is the fluid inside your joints. Chondroitin sulfate, meanwhile, it has this polymer, and it's found in places where you need lots of structure, like cartilage, tendons. Your heart valves have a lot of chondroitin sulfate. Then keratin sulfate, which is not the same as keratin. Keratin is a protein, but keratin sulfate is found in, again, structural places, anywhere you have sort of a lot of dead cells, so bone, hair, and fingernails. And then the cornea has a lot of this material in it. So again, just another structural component of the cell. And then algae and some bacteria also produce this polysaccharide called agarose. It's kind of a complex one because it's this modified version of galactose, which has this ring structure in it. But in any case, agarose, again, for the algae is an important structural element. So like seaweed, for example, which is a form of algae, uses this to support its whole body. But then agarose has some interesting practical applications for humans. So we use agarose in the lab a lot. We used agarose when we did gel electrophoresis. So the matrix that this stuff forms when you solidify it is useful because it lets molecules kind of go through it. And then you can also use agarose as a medium, or agar, which is a version of this, to grow bacteria, for example. So it just sort of sits on top of the solution and grows that way. Okay, one more thing about polysaccharides, and that is that, like proteins and like nucleic acids, they have some secondary structures. So for using the analogy with proteins, the primary structure of a polysaccharide would be just the sequence of amino acids that make it up, and then the way they're connected. And then the secondary structure would just be the whatever shape the polysaccharide folds up into. So just like with a polypeptide, the bonds between monosaccharides within a polysaccharide, those glycoside bonds can rotate, and they can rotate if there's two carbon-oxygen bonds. Then you can have two sort of rotational angles, and so in peptides we call those phi and psi, and we use the same letters for talking about the rotational angles of the glycoside bonds. And that's what determines most of the secondary structure, is what angles they end up as. Except, of course, if you have a situation like Dextran, where the linkage between monosaccharides is, in this case, between the 1 carbon of a glucose and then the number 6 carbon of another glucose. Because the 6 carbon kind of sticks out of the plane of the ring, there's actually a third rotational angle. So the secondary structure can get a lot more complicated with polysaccharides than it can with polypeptides. This kind of, but it does matter what those angles are. So this, for example, is showing 2 monosaccharides kind of in 3 dimensions, and 2 different sets of phi and psi angles. And so one set of angles, you have the 2 monosaccharides kind of laid out in a more or less planar conformation, and here the 2 planes are roughly kind of perpendicular to each other. And here you can kind of map out, for any pair of monosaccharides, you can map out the different rotational angles, and then there's different amounts of interference that are created by different angles. So over here, these two, this is a pretty stable configuration, because the 2 monosaccharides aren't really interfering with each other, whereas over here they're brought closer together, so there's a lot more interference. And so over here you can kind of see basically the stability of different angles. So this configuration for these 2 monosaccharides happens to be more stable, and so it's kind of at a lower energy level. But what it means is there's a really diverse sort of landscape here of possible conformations. And so the three-dimensional structure of a polysaccharide can vary quite a bit for any given combination of subunits. And that's something that makes them different, again, from proteins, because a protein has a defined number of amino acids and a defined sequence, and that sequence determines the secondary and tertiary structures of a protein. And so there's fewer conformations and fewer stable conformations for a given protein than there are for a given polysaccharide. So so these are just the things that affect that. You can see things like helices. So for example, amylose can form basically this is actually a left-handed helix, or cellulose, where again, because the individual monomers within a chain form hydrogen bonds and then can also form hydrogen bonds with adjacent chains, it forms these very sturdy fibrous structures. And so again, it's the secondary structures do affect the structural properties of these molecules, but and it can to some extent affect how they interact with other molecules also. So next time we will talk about glycoconjugates.